There we go. This is the recording for Biology 105, and it is September 17th, 2025, right? All right, cool. Here we are. Yeah, I think so. We're at week 4. That's like a quarter of the way through the semester. You're doing great. Just keep up the good work, all right? Here we are. It's Wednesday. That means that tomorrow you've got your lab sections. I have requested that the lab instructors please have your carbohydrate graphing assignments all graded by the end of the day today so that you'll be able to look at how you did on that and maybe ask them about, why did I miss this point, if you'd like to, in lab tomorrow. Okay, great. Lab this week is a protein assay. Make sure that you access the laboratory materials in the week's module. There's an assignment where you have to analyze data in this file right here. All right, there's the protocol. You'll get an in-lab PowerPoint from your lab instructor that runs through the lab thing. We're doing a colorimetric assay today. And in a colorimetric assay, the protein that you're going to be quantifying is itself colorless, but you're going to be detecting it with spectrophotometry. So what you do is you react the protein with something else, and that makes a product that has color. And then so the amount of that product, which you can detect with spectrophotometry, serves as a proxy for the amount of the colorless substance, which you could not detect with spectrophotometry. Woo, very cool. You've done colorimetric assays already because you already did the carbohydrate assay. All right, but here's another one. It's going to be cool. You get to use the 96 well plate reader. It's a lot of fun. Don't forget to watch the video beforehand. Hopefully this lab goes, doesn't extend beyond the lab period. I really hope that it goes smoothly for all of you. Have a great time. Don't forget, hey folks, guess what happens on Monday? Big old exam, all right? It's very exciting. 25 multiple choice questions worth 3 points each. That means that for each question, you've got 2 minutes to answer it, right? That's based on, what I was taught back when I was learning how to teach. That's like good for multiple choice. You need to give students about 90 seconds to answer them, so, it's multiple choice. Everything that's on the exam is right here in the study guide. Any questions, please? Yeah, no problem. Just remember, C, it's all C's. I just did C top to bottom. Yeah. Yeah, don't forget. That's not true. It's just a fun joke. But I think you would do pretty well if you answered all C's on the exam. You'd get like a 30%, something like that probably. It's pretty good. It's better than 0. I've seen worse. I've seen worse scores. Yeah, negative. So, plus I would laugh so hard, I'd probably give you like bonus points. I'd be like, oh, you put all C's, that's hilarious. You can have bonus points. Don't do that. That's not true. I'm lying to you. I'm just goofing. It's just a prank, okay? All right. So don't forget, exams on Monday, review the study guide, you'll do great. If you struggle with it, if you don't do great, I'll cover the extra credit stuff afterwards, ways to make up points, in case you get 70% or lower on the exam. Any questions? Any questions? Okay. I would love to get into the cell structure and function. Oh, but we're not ready for that yet, are we? I would love nothing more than to be in the cell structure and function PowerPoint right now. But we are still in biological macromolecules. Yeah, yes. All right, cool. Clicker, I didn't plug it in. So, I have laid out the carbohydrates and the lipids for you, okay? And now we're moving to the polypeptides, to the proteins. The proteins are polymers made up of monomers that are amino acids. Amino acids have a similar structure where they all have an amino group and a carboxyl group joined by a carbon, okay? This carbon's called the alpha carbon because it eats a lot of whey protein. Is that a funny joke? It's fun. Okay. So check it out. You join all of these together. You make a long chain. Each amino acid differs in these side chain structures. So essentially, you've got a long repeating chain extending out with a bunch of different groups attached. And those different groups that are attached are going to dictate the folding pattern and the characteristic of the full polypeptide. So check it out. Some of these are hydrophobic, some of them are hydrophilic, some of them have an actual electrical charge, and through the activity of these side chains, the protein will gain a three-dimensional structure, and it will gain some special function. The fact that we have all this different diversity within the amino acids means there can be a ton of protein diversity. That's why they can do so much. Cool? Boom, boom. There we go, there we go. All right. They're joined together by dehydration synthesis, forming a new covalent bond. For these, it's called a peptide bond. What's a peptide bond? It's the bond between amino acids. Don't worry about it, all right? That's why they're called polypeptides, because there's a bunch of peptide bonds. Okay, I don't like this here. Form is function. That's actually pretty good. I can do this and be like, what you don't seem to understand is that the shape of a molecule dictates to a large extent its function, all right? I'll take you out of here. Maybe next time. Maybe next time. So, protein structure, protein form. If form is function, what dictates a protein's form? What dictates its structure? What dictates its function? Let's talk about it. There are multiple levels to protein structure. They're organized from the primary to the quaternary structure. All right, I'm going to explain it all to you. So the protein structure determines its function, and usually a protein is made up of multiple polypeptide chains. So you've got a chain of amino acids here, and it folds up. And then you've got a chain of amino acids over here, and it folds up. And then the two of those associate together. Now they're folded up, and they click together, and that makes the fully functional protein. Okay? So, different levels of protein structure. Primary structure is just the sequence of amino acids. All right? That can be pretty easily expressed, okay? Like in this example right here. Those aren't labeled. I want a labeled one. Right here, okay? Tryptophan, phenylalanine, glycine, serine, that's primary structure. You just need to know the order of the amino acids. Okay? Primary structure is a sequence of amino acids. Secondary structure, okay? Secondary structure occurs because of interactions in the backbone. So remember how the backbone is that repeating like amino carboxyl group with the alpha carbon in the middle, and all of that is just repeated. All right, it's consistent. The backbone structure is consistent, and you're going to see that throughout biological macromolecules. If I say backbone, I mean that, oh, it's a consistent structure, all right, that goes through the whole molecule. So this backbone structure, it's always the same, but it interacts with itself essentially because there's degrees of polarity here. Look at this oxygen, look at this nitrogen. Those are highly electronegative, all right? There's hydrogens in here. So there's actually hydrogen bonding that can occur between the backbone and itself, all right? So the backbone interacts with itself in a repeating way. The structure's repeating, so it tends to generate these very repetitive patterns of folding or twisting. It forms these pleated sheets and these alpha helices. Let me zoom in on this thing here for a moment. So there's the primary protein sequence, is just the sequence of the amino acids in the chain. The secondary sequence, it folds up into these sheets and these helices, all right, based on hydrogen bonding between the backbone and itself. I feel like I'm saying that in a strange way, but are we cool with that sort of general concept? Awesome, I love it. Moving on to the next level, we've got the tertiary structure, okay? The tertiary structure, that's the structure determined by the side chains. So those different projections off of the backbone, where, for example, the hydrophobic projections associate with the other hydrophobic projections. Question, please.
So is there like a general preference for it to be called R groups or side chains?
Ooh, R groups is more general in organic chemistry. Whenever you're talking about a molecule that's really consistent, but it has one specific functional group, side chains is, I believe, it would be more specific to the amino acids. All right. I would say use side chains when you're talking about the amino acids. It's actually a really good question. So the side chain interactions are what determine tertiary structure, but R groups are more just generally when you're trying to show, listen, there's this whole class of molecules and a big chunk of them is the same, but they have a specific region that's different and that gives them different function. All right, awesome. Really, actually, you know what? Very good question, well done. Okay, tertiary structure, great. So it can be all sorts of different interaction types. I'll show you some examples in a moment, but it's all because of the side chains interacting. And then the quaternary structure is when multiple fully folded proteins, okay, proteins that have their own primary, secondary, and tertiary structure associate together, okay? Cool? All right, it could be because this polypeptide here has a bunch of positively charged regions projecting off of its side chains, and this polypeptide here has a bunch of negatively charged regions due to its side chains, and they associate. That's a lazy example, but it should sort of drive the point home, I hope. Question, please.
Is there a quaternary structure?
No. No, there is not, because no matter how many different polypeptide chains associate, it's still quaternary structure, right? So quaternary structure is the top level for protein structures. Yeah. But you could have a protein that's made up of 8 different subunits, and that's its quaternary structure. You see what I mean? Awesome. Awesome, awesome, awesome. Yes, please.
That'd be a weird question, but do proteins prefer a certain structure? Like, they want to reach quaternary all the time, or are they finding like a primary structure?
There are functional proteins that have only tertiary structure, but the tertiary structure is what lets a polypeptide have function. Okay. If it just has primary structure, it doesn't really have much of a function.
Reach at least tertiary, and then optimally it reach quaternary. I don't know.
Yeah.
If I'm saying that. Sorry.
No. I always try to move away from like, oh, it wants to do this, but I say that all the time too. Like, it wants to do this. The water wants to move in this direction. The water doesn't want anything. The protein doesn't want anything. But as a natural outcome of its primary structure, it will fold up into a tertiary structure, which is really what lets a protein have a function, is the tertiary structure. So it's functional at this point, all right? It's also going to have secondary structure because whenever you have one of these backbone chains, they make hydrogen bonds. It builds up. Yeah, it builds off. Now, of course, no problem. Now, of course, all these interactions could be disrupted if, say, you put a protein in something that was super acidic or basic. It could cause the protein to lose its form and therefore lose its function. Who's willing to share the term we use for that process where you cause a protein to lose its form via extreme temperature, extreme pH, etc.? Yes? Denaturation. Very good points. Also, awesome, solid. So we'll talk about denaturation in just a minute too. Okay, questions about different levels of protein structure? Please. I'm just throwing points all over today. You're getting some for that one. So check this out. Peptide bonds are only in the primary structure. And peptide bonds are covalent bonds. And if you remember from that table about relative strength of bonds, covalent bonds are way, way stronger, like 10 times to 100 times stronger than hydrogen bonds or polar-polar interactions, things like that, right? So the primary structure has peptide bonds in it. It's very stable. It's hard to denature that and break it back down into amino acids. But it's not that difficult to make these hydrogen bonds break. And it's even easier to make the tertiary structure fall apart because it's not even all hydrogen bonds, which are like the strongest of the intermolecular forces, right? Anyways, does that answer your question? Cool. There's no peptide bonds in the I mean, there are peptide bonds in the tertiary structure in that it's got the chain of polypeptides, right? But peptide bonds don't determine the tertiary structure. It's all determined by hydrogen bonds, polar-polar or non-polar interactions, positive-negative interactions, right, between charged side chains. It's all side chain stuff. Cool? And none of it's covalent. Awesome. Except maybe disulfide bridges, but let's not worry about those. Why would you bring up disulfide bridges? What are you doing? Are you trying to mess me up? Okay. Primary structure, sequence of amino acids. This is the same thing with a different image to just try to, help cement the concept. Cool? Glycine, proline, threonine, glycine. Like that's the primary structure. All right? Secondary structure, the side chains form hydrogen bonds. All right? Sometimes that makes a helix. Sometimes that makes a pleated sheet. When it does which is determined partially by other proteins and the conditions surrounding the protein when it folds. It's complicated. Yes?
Wait, you said the side chain forms hydrogen bonds? I thought it was
the backbone. It's the backbone. Yeah, the hydrogen bonding. Now, one of the side chain interactions is like a hydrogen bond, but the hydrogen bonding is here in the backbone. Let's just keep it with that. Sorry, if I misspoke, I meant to say the secondary structure occurs due to interactions between the backbone. If I said side chain, I am so dang sorry. Yes.
I'm starting to get it.
Good.
What is it all called? The primary structure, that's just the chain, which is pretty much the backbone. And then the secondary structure, that's like everything coming off of the backbone, perpendicular or helix shaped. Like all of those molecules.
Well, the primary structure, here's the problem. What makes each one of these amino acids different is the side chain. So this is all just backbone as far as the bonds that are forming. I think this is what you're saying. I just want to make sure that I repeat it to make it clear.
Oh, well, in that case, is the secondary structure, let's say multiple backbones and amino acids basically side to side forming swirls because of their hydrogen bonds.
It's basically that the backbone is interacting with itself. Okay, so if my arm is the sequence of amino acids and I have a bunch of these side chains projecting off of it, and this portion is the backbone, then it might form some hydrogen bonds by interacting with itself.
Each group's own intra and intermolecular forces like hydrogen bonds.
Yeah, so
certain charged atoms and molecules will try to orient itself in a certain way. Same with saturated and unsaturated fatty acids.
kind of, but I think that bleeds too much into tertiary structure. So with the backbone, what's going on is because of the intramolecular forces in the backbone, right? You end up with degrees of polarity of the backbone. So the backbone has polarity, and therefore it can interact with itself and form hydrogen bonds like this to make, say, a pleated sheet, which is like an accordion.
Now I get it.
Does that make sense?
How everything interacts with itself and
Yes.
how it gets its shape.
Yes. Oh, awesome. I'm really happy to hear it. Yes, hand in the air, yes. Oh, sorry. Oh, no big deal. All right, yes.
I just wanted to quickly get a refresher. The hydrogen bonds are made because of the polarity.
Yeah, because of the polarity between and I'm really happy that honestly we're getting some really good questions today. I'm very happy to talk about this. The bonds here between this nitrogen and this hydrogen and these hydrogens here, that's going to be a very polar covalent bond because nitrogen is way more polar than hydrogen. So that's going to mean that there's a partial negative charge here and a partial positive charge in this portion of the backbone. And to a lesser extent, the same thing will happen here. So there's going to be regions of polarity throughout the backbone. And therefore, the positive and the negative patches interact with each other and they form like the pleated sheet. The pleated sheet's the only example that my arms are capable of doing, because of how arms work. Good deal. Okay, primary, secondary structure. Now, this spider silk thing it's like a chance for me to talk to you about how micro form and macro form are both like similar. It's all physics. You think about like an accordion stretching out and how that could withstand a lot of tension. And then you think about that same thing with the pleated sheet structures and how if they stretch out, they could withstand tension. So having a protein making up a spider that has a bunch of pleated sheets, it's similar, all right? But this is a more of a like, I'm gonna blow your mind point that doesn't usually blow any minds. So, hey, here's what you can take from this. Spider silk's got a bunch of proteins in it. All right, it is made of proteins, okay? And it's got a bunch of beta pleated sheets in its structure, and that alone gives it a lot of its elasticity and stability. Cool. I'm not gonna quiz you on spider silk. But you could point out, like, did you know that's a protein? You know, which is kind of weird to think about. Okay, tertiary structures, ready? Side chains are interacting. So, to get at your point earlier, there are hydrogen bonds that can occur between the side chains. But there are also all of these other side chain interactions, and many of them are weaker than the hydrogen bonds, and they're not as consistent as those backbone interactions. So the backbone interaction is consistent and is pretty stable, meaning secondary structure ends up being more stable than tertiary structure. So what I'm trying to hint at here is, as a protein begins to denature and lose its form, the first thing it's going to lose is its quaternary structure, because this is fully formed molecules that are just sort of held together, right? Those are going to fall apart. The next thing it'll lose would be its tertiary structure, because some of these interactions are pretty weak. Van der Waals, hydrophobic interactions, the disulfide bridges are pretty strong, the hydrogen bonds are pretty strong, but, you know, they're not as consistent in their strength, so that'll fall apart next. Then the hydrogen bonds start to fall apart, all right, between the backbone and itself, and you lose secondary structure. Only under very, very extreme circumstances will you actually lose primary structure, because primary structure is all maintained via covalent bonds, those peptide bonds between the amino acids. All right, so it all comes back to discussing relative stability of bonds, and that'll determine how proteins denature. I know it's a lot, but I'm hoping that you can see, like, oh, well, it all built up to this, from our chemistry review all the way to understanding the structure of a protein. Either way, that's the hard part about any of the sciences, is they all work together and everything is translatable to the other fields. Like every single thing you learn applies to every other thing, but it's so hard to draw the connections. You know, that's the difficult piece. Anyways, sometimes you don't want to draw all the connections when you're focusing on passing a class, and that's okay. Some people don't like biology as a topic, and that's okay too. You like other stuff. I don't know, something cool. You like skateboarding or something, I don't know. But that's okay too. Anyway, check this out. Here's collagen. Collagen's like a fiber made up of a few different types of proteins that are all wound together and woven together, and they got a bunch of these helical structures. Lets it be pretty stretchy. All right. Here's hemoglobin. Hemoglobin has different subunits that are specific, right? There's got these alpha and these beta subunits. Each one of these is a protein with primary, secondary, and tertiary structure. The alpha subunit is different from the beta subunit. They're joined together to give a quaternary structure to hemoglobin, which then also uses heme, a molecule containing iron, in order to actually carry the oxygen through your bloodstream, for example. Yes, please.
So is this showing that an alpha helix can bond to a beta sheet and quaternary?
Oh, thank you so much for bringing that up. This is called an alpha subunit, but that's different from the alpha helix and the beta pleated sheet. It's just a naming convention. If you have a protein with multiple subunits, you'll call it, this is the alpha subunit, this is the beta subunit. It has nothing to do with the alpha and the beta. You've 100% got it. They'll both have both. Isn't that good?
Wait, so alpha can bond with beta?
I mean, it depends on what you're talking about, the alpha helices and the beta pleated sheets. Not really. Yeah. No, this is a bad path. We need to pull away from this path that we're walking down right now because it's just too rocky and it's all misconceptions. I know, I totally hear you. So what we're gonna do is gonna dial it back. We're gonna put a fence up and we're not going down that path, okay? So, alpha helices, beta pleated sheets. Those are two different structures that occur because of backbone interactions. You could have an alpha helix that leads into a beta pleated sheet in a protein, and then those could be folded to be in association with one another within a protein. Within a protein. Now, when we're talking about a protein that has quaternary structure, we'll call some of the subunits alpha subunits, some beta subunits, some gamma subunits, all right? But those Greek letters are just there to identify, like, this is the alpha subunit of hemoglobin. The different subunits. Yeah, it'd be like, this is subunit A, subunit B. That's all we're saying. Yeah. The fact that it's alpha and beta is just an undesirable outcome here.
But helix can be with sheets.
No. So in a protein, right? Let me have a good picture. So check this out, right? Like here's human growth hormone. And you'll see how it will have helices in it. And it will have beta pleated sheets in it. They're not diagrammatically represented as well on this, but say that's a beta pleated sheet. They're not necessarily linked to one another, but they are coexisting as part of the protein structure.
They are coexisting
In the same protein.
Okay.
Is that good?
Yes.
Okay, cool.
They're not like together. They're just like
Yes.
like neighbors.
They're like neighbors.
In the same house.
Yeah.
Okay.
Yeah. Awesome. We got there.
Thank you.
No problem. And it's really good to work through these things because, like, it's really confusing, especially when you have overlap in terminology. So I hope it didn't come off as I was frustrated with you. I was frustrated with the situation. It did. Okay, well, I do genuinely apologize. Okay, thank you so much for accepting my apology, and I will do my best to make amends moving forward. Very good. Cool. Yes. Sorry. I know, that couldn't have felt good. I'm so sorry.
What does alpha and beta signify when it comes to like the differences between the two?
A naming convention, right? Yeah, like it's better to think of them as pleated sheets and helices because that actually tells you something, right? But we call the pleated sheets are the alpha pleated sheets, all right? And the beta helices are called the beta helices. But the important thing is pleated sheet versus helix. But then for us, not even that's important. The important thing is that both of these structures result from backbone interactions, whereas the tertiary structure results from side chain interactions. Yeah. A solid. Not a satisfying answer, I'm fully aware. Yeah, yes, please.
So, how are we supposed to tell the difference between the secondary and tertiary structure if they're both like the same look, and you're just supposed to be looking like inside and they're different?
Absolutely. So to tell them apart, right, you would get, in my class, you would get a description of what led to the structure. That's what tells them apart. It's not that you would like see a picture of a protein like this, and I'd say like, highlight the part that is leading to tertiary structure. It doesn't really, work that way. It's more a question of what force is leading to the structure. Or what bond is stabilizing the structure, or what interaction is stabilizing the structure. So with the primary structure, it is specifically describing the sequence of amino acids. It doesn't really tell you anything about the folding pattern or the shape at all. The secondary structure tells you something about the shape, all right? But it just tells you, does it have these twists or does it have these accordions in it? And it's all due to the backbone interactions. The tertiary structure gives you like a globby shape like this, because the side chains start to interact. Yes, please.
So wait, the side chains are the connections like between carbons? Like how is that?
Let me show you here. right here. So remember, right, in these amino acids, you've got the repeating unit of the amino group and the carboxyl group, or yeah, the carboxyl group, which here has the hydrogen missing to show where the bond will form. Don't worry about it. All right? But so you've got right here, every amino acid has the same structure here, and it's different on this projection. This is the side chain, okay, right here. So when you look at the tertiary structure of a protein, this is indicating the backbone, the purple line, and you've got these side chains projecting out. And these side chains will interact with one another, leading to a specific folding pattern for the protein. It also will make pockets on the protein of, say you've got a bunch of acidic side chains in a pocket on a protein, well now you've got a little acid chamber. And if you wanted to break something down, you could put it in there and then go and like melt it. You see what I mean? So these side chains are very important in determining specific function of the protein.
So on a secondary structure, would you see any side chains, or is it just that like the side chains are all like the same thing, just multiple instances of those?
The secondary structure is determined by the backbones. There are side chains projecting off of this. Every amino acid has a side chain, every single one. But the side chain isn't involved in determining the secondary structure. It's just the backbone. The tertiary structure is what's determined by the side chains. Yeah, because each separate polypeptide has its own, like, it's a long strand. It's got a backbone with two ends. And then it folds up and gets a three-dimensional structure. And then there's another one over here that folds up and does the same thing. And then they just click together to make quaternary structure. How did that work for you? Is that okay? Better? Okay, cool. Awesome. Okay. So, folks, let's look here at how structure affects form, affects function, with the example of sickle cells, leading to sickle cell anemia, this concept here, right? So, there's a specific genetic sequence that encodes the beta subunit of hemoglobin, all right? If there's a mutation in the genetic sequence that encodes the beta subunit, you get a different primary structure. You get a different sequence of amino acids. That different sequence of amino acids, that affects what side chain is being exposed here. And that is going to influence the tertiary structure of the protein. Also the secondary to a small extent, but let's not panic too much about that, because remember, the backbone's still the same, all right? So the side chain is going to be the primary thing that's changing this actual structure here. So the normal beta subunit looks like this. The mutated beta subunit looks like this. And it leads to an exposed hydrophobic region on the beta subunit. Now, when associated with the alpha subunits to make hemoglobin, normal hemoglobin looks like this and has no exposed hydrophobic region, okay? Sickle cell hemoglobin, however, has these exposed hydrophobic regions. And what do we know about hydrophobic regions? Like dissolves like, right? They're going to associate with other hydrophobic regions. So these sickle cell hemoglobin molecules associate with one another, and the hydrophobic regions just sort of link up. Because water's buffeting them around, you know, and they're going to just associate together. Eventually, it leads to a more stable structure. And that crystallizes into this fiber. You get that crystallized hemoglobin, you end up with a red blood cell that has a specific sickle shape. All right? It's kind of interesting. So a change in a single amino acid due to a change in maybe a nucleotide or two in your DNA, and now all of a sudden you've got a different primary, secondary, tertiary, and quaternary protein structure, which leads to a very different behavior of the macromolecule, leading to a very different shape for an entire cell, leading to a very different phenotype for human beings that have these sickle red blood cells. All right? These can then further sort of hook up and get stuck in your blood vessels. They cause some problems. It also is hard for malaria to infect these blood cells. You've probably heard of this as an interesting example of positive and negative selection both working together, where if there's no malaria present, this isn't very adaptive. But if there's a high pressure of malaria in an environment, then this becomes adaptive and therefore it reaches higher, prevalence in a population. It's fascinating. But anyways, that's all our stuff about protein structure. No, it's not, because we're going to talk about denaturation. We're almost done with protein structure, I promise. So you ready? Let's keep on going. I talked to you already about acids and bases, okay? Water dissociates. It's a little bit reactive, okay? So there's always some amount in water of hydrogen ions, and there's some amount of hydroxide ions in a solution of water. But if they're equal, then the water is usually water, and it's not charged particles. If you have a basic or an acidic solution, you have an excess of one of these ions, and what that does is it makes the solution have a bunch of charged particles. Cool so far? I love it. All right? We've talked about pure water and the pH scale and what it is and how to calculate it and blah, blah, blah. You probably don't want to hear it all again. Now, check this out. If there's a bunch of charged particles, then the side chain interactions can be disrupted. And the hydrogen bonds that make the secondary structure can even be disrupted. So effectively, I'm not thrilled with that. I'm going to actually just I'm going to use the example here. This is better for me, okay? Check this out. This oxygen right here is going to have a partially negative charge, okay? Ionic bond. This is the one we want to look at. Check this out. So let's say you put this protein into an acidic solution. Well, all of those positive ions can surround this negative charge and neutralize it, okay? And if that gets neutralized, there's nothing for this side chain to interact with. So now this isn't held together as tightly, and it loses its shape, and it unfolds, okay? Same thing with like the hydrophobic interactions and the hydrogen bonds. They can all be disrupted with different pHs. We're not going to worry about isoelectric points. I tried to do it last spring, and it just ended up being more confusing than it was worth. So don't worry about isoelectric points. All right. So denaturation, what is it? It's when a protein loses its secondary, tertiary, or quaternary, tertiary, secondary structure. Yes, please.
Is this when meat changes shape when you cook it?
Yeah, that's protein denaturation. So denaturing proteins, you heat them up until they lose their structure, and then they fall back to another stable state usually. But protein denaturation is a way to make food more digestible. That's one thing that it does. It's not the only reason we cook things, right? But it's one benefit of cooking things, is you denature proteins and it makes them more digestible. Which one? What now? Oh, yeah, you got me. Yeah. Oh, yeah, totally. So you can do it with lime with an acidic solution, too. It's the same net result, where you disrupt the quaternary, tertiary, and secondary structure. The question is just, do you do it with heat, like with egg, or do you do it with acid, with an extreme pH, like in ceviche, right? I always tell this story. It seems to work. I was a child. Think back to when I was a child. Remember me. Here I am. I'm small. I'm young. I live in a town. There are 900 people in this town. Roslyn, Washington, where coal was king. We have nothing to do as children. One day, one of my friends says, "Hey, John, my dad's taking us on a train. It's going to be a wine and tea train, and we're going to go someplace." I'm like, "Cool, a train? I've never been on a train before. I'm a child. I live in the dirt. I live in the filth, So I get on this train, and they say, "Here's the tea service," and they put tea in front of me. And they say, would you like milk or lemon? And I say, yes, please. That sounds lovely. And honey? Yes, I would like that as well. Thank you. I get all these ingredients in front of me. I want to be fancy. I have no culture. So what I do is I take the milk and I put it in and I stir it up. And then I take the lemon and I put it in and I stir it up. And then I take the honey and I stir it up. And I look in my teacup and what I've got is essentially like a cottage cheesy substance inside. It's hot. That hot cottage cheese with honey in front of me. Why? Because I exposed milk proteins to a high temperature and an extreme pH. Now, not wanting to make a scene, I drank the entire cup of tea. And, I never made that mistake again. But that's an example of denaturation where the protein's pretty stable. The milk protein, like you can heat up milk, have a cup of warm milk, and it still is like milk, right? But you throw some acid in there, it's like from lemon juice, and boom, you get it to denature in a nasty way. Fun. It's a fun fun little story, I think. Drew McCoy. That was my friend. I don't remember his father's name. He's not doing well. But that's okay. He's gonna make it through, I believe. No. He's just, it's small town drama stuff. Everybody, all the friends fight all the other friends and you go through stuff with them. It's great. Every time I go back and visit, it's like, who's in a fight with who right now? Why am I telling you this? Because I'm just trying to engage with you and have some fun. And also, I've been talking for 45 minutes and, this is difficult. I might make this look easy, but oh my God, it's so difficult. Thank you. Wait, make it look easy? Okay, that's a bad sign, I think. I need to make this look like it's more work so they pay me more. Oh, everybody, let's talk about nucleotides. Yeah, please. Yes, it can. Thank you for bringing that up. It can. It's called renaturation. It's usually accomplished, it can happen in a cell with these other proteins that are actually called like chaperonins, like a chaperone. And it's like another protein that surrounds a protein and sort of nurses it back to health. It makes conditions that are amenable for it to restructure. So there are ways to renature proteins. The question of whether it'll happen naturally is all about how denatured did it get. Awesome. I love it. This is great. Protein denaturation's all around you. Good. The last group, we're on to nucleic. Oh, we're doing great. It's Wednesday, though. If this was Monday, I'd be stoked. We'd be doing great. We're a day behind. We're fine. Nucleotides. Let's talk about them. Nucleotides also follow the polymer-monomer framework, and just like the polypeptides, just like the proteins, they have a repeating backbone region. All right? It's called the sugar phosphate backbone. Look at this sugar. Look at this phosphate group. It's going to be a bunch of repeating units of sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, making up the sugar phosphate backbone. And projecting off of each nucleotide will be a slightly different nitrogenous base. So it's very similar to the side chains in the amino acids, except that there's only 4 different nitrogenous base side chains here. And we're not going to be really trying to develop a bunch of polynucleotides that have different shapes for function. You want to make a really stable structure. All right? Sick. So, what's a nucleotide? A nucleotide is the monomer of a nucleic acid, also called a polynucleotide. Really what we're talking about here with the nucleic acids is DNA and RNA. All right? Those are the 2 things that we're talking about, the nucleic acids. So, the building block is the nucleotide. It's got a nitrogenous base, a sugar, and a phosphate group. Okay? Nitrogenous bases, sugars, the structures. I really don't think it's reasonable to start this right now, so we will get into it. It's only like 11 slides. We'll do the nucleotides, the nucleic acids on Friday. We'll be ready by the time the exam hits because we'll get into cell structure stuff. We'll be fine. We'll be fine. Have a great day, everybody. Thank you for making it to another Bio 105 lecture. And would you prop that door open on your way out? Thank you so much. See you later. Thank you.